Hello and welcome to the 2017 edition of the RSNA meeting in Chicago. My name is Brian Casey. I'm editor-in-chief of AntMini.com. So here at this year's RSNA meeting, there are some pretty interesting experiments in human perception going on over in the Lakeside Center. Dr. Jeremy Wolfe is with Brigham and Women's Hospital, and he's going to be talking about what's going on over there. Dr. Wolfe, thanks for being with us today.
Thank you very much, Brian. This is the second year that we've been running a medical image perception lab here at RSNA. This is a project funded by the National Cancer Institute. And, well, you might wonder why in the world it's interesting to do visual perception experiments at RSNA, but I think if you ask almost any radiologist, you would find out that they say, look, sometimes I just miss things that are in that image, and if you look at them later, it's absolutely clear that it was there. Yeah. And we'd really like to understand how that comes to be. Why is it that people miss things that are clearly visible in front of them? And in the case of something like radiology, what could we do that would make this better? So what we've been doing, there are a number of labs around the world that work on these sort of problems, and for the last couple of years, we've brought I guess there are 14 different labs from at least 3 different continents this year who've brought their research to RSNA because RSNA is where the radiologists are. It's a lot easier to test radiologists when they have a bit of free time walking around at RSNA than it is to try to pry them loose from the reading room back at their hospital.
Right. Now, I was over at Lakeside Center yesterday, and I noticed that you've got a number of. It's in the posters area. So if you're going over to Lakeside and you're looking at posters, you'll see a bank of looks like about 5 or 6 rooms with dark curtains and a desk in there and a monitor and a light, and so it looks a little mysterious. What sort of things are happening over there?
Well, we've got a variety of different experiments running at the moment. So there is one lab from California that is tracking the eyes of mammographers while they look at breast cancer cases. We have a lab from the National Cancer Institute interested in quantifying prostate lesions. Barco has a monitor where they want to figure out if their color rendering is the right color rendering. So it's a range of different perceptual issues that we're investigating. We can happily test radiologists who are general radiologists, and we'd be particularly delighted, I think, at the moment to see people who read quite a bit of mammo or chest CT. We have a couple of chest CT ones running. And these are experiments that take anywhere between 10 minutes and a half an hour. One of the other things that we've discovered is that even at RSNA, we can't recruit radiologists to sit in a dark room for 2 hours with us, so we try to keep it short.
Okay, and so anyone can just walk right up and check in and be sat at one of these experiments?
Yep, absolutely. We'd be delighted to see people anytime. We're here from 8 to 4 every day during RSNA.
Okay. Now, you also did this last year. What were some of the things that came out of last year's research?
So, I can talk a little about the experiment that my lab was running. What we found in some earlier work was that if we presented mammograms for half a second and asked radiologists to classify the mammogram as normal or abnormal, after we got them comfortable with the idea that they could do this, they could even answer the question in half a second. It turns out that radiologists can do that. They're not perfect, but they comfortably beat chance. And last year at RSNA, what we were doing was showing mammograms from women who had not yet developed cancer. So we went back, took the priors from 3 years earlier, flashed those for a half a second, and it turns out that even 3 years before the cancer is there, there's something in the image that says abnormal. And now we're pursuing the possibility that could be some form of a useful imaging biomarker. But the hardest thing to do in this kind of research is to get enough radiologists into your experiment so that you know that you've got something that's statistically reliable. And that's the really great strength of RSNA, because there are a few radiologists here.
There's a few. All right.
We think.
Yeah. Well, get on over to the Lakeside Center. It's on the main floor, I believe, in the poster area.
Out back by machine learning.
Yep, back by the machine learning section. So get on over there, participate with some of these fascinating experiments. Dr. Wolfe, thanks for being with us today.
Thanks very much.
All right. Signing off for AntMini.com, my name is Brian Casey.